Good day, everyone. I'm Sir Franz, and I'm welcoming you to our lesson on the pentose phosphate pathway. This will be our topic outline. The pentose phosphate pathway has another name. It's also called the hexose monophosphate shunt. It occurs in the cytoplasm of cells, and it is a minor pathway for carbohydrate metabolism. And the major pathway that we are referring to here is glycolysis. It specializes on synthesizing products that the cell needs for biosynthesis, or, in easier terms, anabolic reactions. The two main products are reduced NADP, or simply NADPH, and ribose and other sugars. So this illustration on the right is quite simple and important because it shows us that glucose 6-phosphate from the phosphorylation of glucose catalyzed by hexokinase can either enter the major pathway, which is glycolysis, shaded here in light blue, or it can be shunted into the minor pathway, which is the pentose phosphate pathway. So the pentose phosphate pathway shown in this image is colored green and purple. The green portion of the pathway is also known as the oxidative phase, and the purple portion of the pathway is known as the non-oxidative phase. The two main products of the pentose phosphate pathway are ribose 5-phosphate and reduced NADP or NADPH. So, you will now understand that the name of the pathway tells us of what the products are. Pentose phosphate pathway refers to the product which is ribose 5-phosphate. Pentose, that's the ribose, and the phosphate is the phosphate attached at carbon number 5 of ribose. Ribose 5-phosphate is the precursor for the synthesis of nucleotides, which will be our future lesson. But suffice to know that nucleotides are the building blocks of genetic material. For example, RNA and DNA. The simplest monomer of those would be nucleotides. For us to synthesize nucleotides, we need a backbone, some form of a scaffold, which is the ribose 5-phosphate, a form of highly energized sugar. So the next product would be reduced NADP or NADPH. It is a redox coenzyme, which means it participates in reduction oxidation reactions. It is used for reductive biosynthesis of fatty acids and cholesterol. The term reductive biosynthesis simply means that we are performing an anabolic reaction, anabolism, with concomitant reduction. So that means for us to build larger molecules, we need to reduce these molecules. And the hydrogen that you need for reduction is supplied by NADPH. NADPH is also used for the defense against oxidative damage. So remember that in the cells, because of the reactions that occur within the cell and in the cell membrane, there is production of free radicals. More so because we are utilizing oxygen in our redox reactions. So these free radicals can damage the cell, and they can even form chain reactions. And so the cell has to find a way to truncate or to stop the free radical chain reaction, and that is through the use of glutathione. Here, in this image in the right, you will see how NADPH performs its antioxidant property. So you have here the pentose phosphate pathway, which produces NADPH. Together with the action of riboflavin, which is in the form of FAD, NADPH donates its hydrogen to the oxidized form of glutathione, which is GSSG. So NADPH gives up its hydrogen to oxidized glutathione to reform the scavenging reduced glutathione, which is GSH. This reduced glutathione, or GSH in the image, is then able to neutralize hydrogen peroxide, which is an oxygen-free radical. So by neutralizing hydrogen peroxide is not able to damage the cell. Furthermore, hydrogen peroxide is converted to water, while reduced glutathione is oxidized into oxidized glutathione. To sum it all up, the NADPH from the pentose phosphate pathway is needed by the cell to reform reduced glutathione so that we have enough reduced glutathione to scavenge oxygen-free radicals. As I have emphasized in the first slide, the pentose phosphate pathway has two branches, an oxidative branch and a non-oxidative branch. The oxidative branch synthesizes reduced NADP or NADPH. The most important enzyme in the oxidative branch is glucose 6-phosphate dehydrogenase. So let's slow down. Glucose 6-phosphate dehydrogenase. So it will catalyze oxidation of glucose 6-phosphate. This oxidation of glucose 6-phosphate is the committing and rate-limiting step of the pentose phosphate pathway. That means when glucose 6-phosphate is already oxidized, the product will not be able to go into other pathways but to the pentose phosphate pathway only. It's called the rate-limiting step because the speed of the entire pentose phosphate pathway depends on the speed and activity of glucose 6-phosphate dehydrogenase. If glucose 6-phosphate dehydrogenase acts slowly, then the pentose phosphate pathway will also act slowly. So this is important because we have a condition known as glucose 6-phosphate dehydrogenase deficiency, where patients have a genetic lesion that decreases the activity and the amount of the enzyme. Thus, these patients are not able to produce lots of NADPH. When they have low levels of NADPH, the functions that NADPH performs will not only be altered in these individuals, but may even be absent. Okay? So let's now turn our attention into the image in the right. At the top, that's the structure of glucose 6-phosphate. So you have glucose and a phosphate group attached to carbon number 6. Through the action of glucose 6-phosphate dehydrogenase, carbon number 1, which originally holds a secondary alcohol, is oxidized into a ketone. And the product there would be 6-phosphogluconolactone. You will appreciate that at carbon number 1 or position number 1, you have a ketone body. Now, because glucose 6-phosphate is oxidized, remember, there should be coupling. So what is reduced? The substance that is reduced is NADP. So when NADP is reduced, you form NADPH. And that is where your NADPH comes from. Okay, let's continue. 6-phosphogluconolactone is then hydrolyzed, and what happens here is that the chain configuration results from the hydrolysis, and you destroy the Haworth configuration. Next, this product is already known as 6-phosphogluconate. Okay? Now, 6-phosphogluconate is further oxidized, again, with a concomitant reduction of NADP to form reduced NADP. But at the same time, because we have an unstable carboxyl group at one end of 6-phosphogluconate, that will be released as carbon dioxide. And the end product here would be a 5-carbon sugar plus carbon dioxide plus reduced NADP. So our 5-carbon sugar here is ribulose 5-phosphate. So let's summarize. What are the two steps that produce reduced NADP? That's the oxidation of glucose 6-phosphate to produce 6-phosphogluconate, and the oxidation of 6-phosphogluconate to produce ribulose 5-phosphate. Ribulose 5-phosphate then enters the non-oxidative branch. The non-oxidative branch will link ribulose 5-phosphate to glycolysis and gluconeogenesis, which is the major pathway. We have two important enzymes in the non-oxidative branch. Transketolase is an enzyme that transfers 2 carbon units, and transaldolase transfers 3 carbon units. Transketolase, which transfers 2 carbon units, utilizes for its function vitamin B1 or thiamine pyrophosphate. Okay, so let's start with the image here in the right. Ribulose 5-phosphate again is from the oxidative branch of the pentose phosphate pathway. Ribulose 5-phosphate, turning to the left, will be acted upon by an isomerase to form ribose 5-phosphate. Again, ribulose is a ketone sugar and ribose is an aldehyde sugar. Ribulose is a ketose, ribose is an aldose. So with the action of isomerase, we have converted the ketose into an aldose. Ribulose 5-phosphate can also be acted upon by an epimerase, which means that it acts only on 1 carbon in the 5-carbon sugar. By the action of pentose phosphate epimerase, we form cellulose 5-phosphate. The two products of isomerase and epimerase, that is, ribose 5-phosphate and cellulose 5-phosphate, then undergo reaction with transketolase. Okay, remember that transketolase, the action is to transfer 2 carbon units. The source of the carbon will be cellulose, and the recipient of the 2 carbons here will be ribose. So, the 2 carbons from cellulose are transferred to ribose, and now ribose becomes 2 carbons longer. The aldehyde ribose, after receiving the 2 carbons, becomes a ketose in the form of pseudoheptulose, which is 7 carbons long. But remember, it's already a ketone sugar. Whereas cellulose, who lost 2 carbons, becomes 2 carbons shorter. And therefore, we have a 3-carbon sugar, glyceraldehyde 3-phosphate, which is an aldose or aldehyde. The next step These two products, the 7-carbon sugar and the 3-carbon sugar, will then be acted upon by transaldolase. Remember that transaldolase catalyzes transfer of 3 carbon units. Okay? So, the 7-carbon unit of pseudoheptulose will lose 3 carbons and transfer it to glyceraldehyde. So by losing 3 carbons, it becomes 4 carbons long, and that is your erythrose, which is again an aldose, and the one that receives the 3 carbons, glyceraldehyde 3-phosphate, which is an aldose, becomes 3 carbons longer, and it becomes fructose, which is a ketose. So from C7 and C3, transfer 3 carbons, we form C4, 4 carbons, and C6, 6 carbons. Okay? Now, fructose 6-phosphate can already enter glycolysis. And glyceraldehyde 3-phosphate, remember, these processes occur in the cytoplasm, where glycolysis also happens. Glyceraldehyde 3-phosphate can be taken away from the pentose phosphate pathway and enter glycolysis. Okay? So that is the non-oxidative branch. The key points here are ribulose 5-phosphate enters, and it can produce ribose 5-phosphate, which is a major product that can be taken away from the pentose phosphate pathway into the synthesis of nucleotides. Okay? So, here in the non-oxidative branch, you have interconversion of sugars so that these sugars can eventually enter glycolysis. Okay? And again, we have two important enzymes, transketolase, catalyzing 2 carbon unit transfers, and transaldolase, catalyzing 3 carbon unit transfers. Now, the pentose phosphate pathway runs in 2 modes, which means the steps that we have discussed can either be modified so that it runs faster or produces a particular product. Again, what are those products that are important? Reduced NADPH, which we need for prevention of oxidative stress, and for reductive biosynthesis, and ribose 5-phosphate, which we need to synthesize nucleotides. So what are the 2 modes? When the cell needs a lot of ribose 5-phosphate, ribose 5-phosphate is formed not only through the oxidative branch going down, but also ribose 5-phosphate is formed by the reversal of the reactions from the non-oxidative branch. So you have reactants going up the pathway. Okay? And as you have observed, glycolysis is linked to the pentose phosphate pathway. So you can even take away reactants or intermediates of glycolysis and feed them through the pentose phosphate pathway to produce ribose 5-phosphate. Again, the first mode, when you need a lot of ribose 5-phosphate, the oxidative branch proceeds faster and you reverse the non-oxidative branch to form ribose 5-phosphate. This happens when the cell is actively synthesizing genetic material, which again requires nucleotides to be formed. So nucleotides come from ribose 5-phosphate, or ribose 5-phosphate is important for the synthesis of nucleotides. So this happens when the cell is in the S phase of the cell cycle, where you replicate DNA. When more NADPH is needed, so the other major product of the pentose phosphate pathway is the focus here. You need a lot of NADPH, for example, to scavenge more free radicals or to produce fat and cholesterol. So that's the time when you speed up the production of NADPH. So what happens? The two branches, the oxidative, proceeds faster, and the non-oxidative proceeds also faster to produce eventually fructose 6-phosphate and glyceraldehyde 3-phosphate, which then feeds into gluconeogenesis. Then it goes up until you produce glucose 6-phosphate or glucose. Through gluconeogenesis and the production of glucose 6-phosphate, you then shunt the glucose 6-phosphate back into the pentose phosphate pathway so that it is oxidized to produce reduced NADP or NADPH. Okay? So here are my references. And through this lesson, we have described the pentose phosphate pathway. It's a minor pathway and it's connected to glycolysis and gluconeogenesis. And the two major products are NADPH, which you use for protection against oxidative damage and production of fats, and ribose 5-phosphate, which you use to produce nucleotides. Okay, so the pentose phosphate pathway has two branches, an oxidative and a non-oxidative branch. So thank you for listening.